Good afternoon to everyone. A very warm welcome to the sixth day of the Faculty Development Program and recent advances in novel drug delivery applications, which has been going exceptionally well. It has been a great pleasure to me to introduce today's resource person, Dr. C.V. Venkata, who is a respected authority on scientific officer grade 4, Liquid Dielectricals Laboratory, Central Power Research Institute, Regional Testing Laboratory, Noida. He pursued his PhD on Polymer Science Technology in 2010 from SK University, Anantapur. Sir research focuses on synthesis and characterization of polymeric matrices for controlled drug release and pervaporation applications. He is a recipient of Meritorious Fellowship UGC New Delhi and Brain Korea for 21 prestigious fellowships at South Korea. Sir published a book entitled Vegetable Oil Based Biolubricants and Transformer Fluids in Springer Nature, Singapore. He has one patent on green insulating fluids for transformer applications, a non-edible vegetable oil. To add to these credits, he published over 32 international journals and national journals. Sir attended 11 national conferences and 9 international conferences. Sir is passionate about solid and liquid insulators for transformer applications, polymer brain composites, and polymeric membrane for fuel cells and pervaporation applications. He has worked as investigator and co-investigator in IHRD project with innovative titles and high outlay. Sir's research experience has played an integral part with several prestigious positions like Scientific Officer Grade 2, Grade 3, Grade 4, and in Bangalore and Noida also, as postdoctoral researcher and research professor in Chengwon National University, South Korea. Presently, Sir is working as Scientific Grade 4 at Central Power Research Institute, Regional Testing Lab, Noida. With a passionate for innovation and excellence, Dr. C. Venkata Prasad has established himself as a leading person in the scientific field. We welcome you, sir, and request you to take over the session. Thank you.
Thank you very much, madam, for your nice introduction. So coming to the today's session, in my previous presentation, we have discussed about the role of importance of polymers and the role in biomedical applications as a control drug release systems. So today we will be discussing about the mechanisms in that various types of control release systems and discuss about the targeted drug delivery system, ocular drug delivery system, and mucoadhesive drug delivery system, and current research scenario and future directions of polymers in drug delivery application. And finally, we will be concluded with some conclusions. So coming to the mechanism of the drug release, so wherever the drug imbibed in the polymer matrix, there will be some mechanism how depending upon the properties of the polymer or drug properties. So during the drug release through polymer matrices, some of the parameters we have to keep essential while designing the control sustain release systems for various drug delivery applications. And these mechanisms control how the drugs will be delivered to the targeted site by through polymer based carriers and other carriers, and they also be tailored to achieve a specified targets release profiles, either may be slow, depending upon the requirement, it is slow, sustained, or burst release. So slow release in case of prolonged this one chronic diseases cases, we have to make the design in such a way that the drug release should be slow and sustained. Whereas suppose if you are suffering with acute problems, then immediate reaction should be get in addition to the in order to achieve that one, you have to make the formulations as per burst release mechanism. So the release mechanisms are primarily influenced by the properties of the polymer, whether it is hydrophilicity, whether it is biodegradable, or it is a cross-linking polymer, or the type of the drug, and the environmental conditions such as depending upon the pH, because in the body there will be different environmental conditions depending upon the area of the site, and also temperatures. Suppose if the patient is suffering from fever, the temperatures also will vary, and depending upon that illness, the various sites will be having various temperatures, and depending upon the ionic strength also, these release profiles will be depending. These are the mechanisms we are going to discuss primarily from the polymer matrices, how it will be the releasing of drug can be observed, such as diffusion and dissolution control system, swelling control release system, matrix erosion control release system, osmotic control release system, ionic exchange control release system, stimuli responsive release system, which is also can be called as a smart release system, and physical or mechanical disruption, also called as burst release system. Coming to the first mechanism, that is diffusion control release system. If you see this two systems are there. One is diffusion control system and dissolution control system. In case of diffusion system, we will be using insoluble polymer, whereas in case of dissolution system, we will be using slowly soluble polymers. Here, we are imbibing the polymer in a system polymeric matrix, and then it is expressing with the time, it will be diffusing, because the concentration in the environment and concentration in the drug formulation system is different. Because of this variation in the drug and concentration, so it will diffuses through polymer matrix when in contact with the body fluids. And after some time, suppose if it is one time it is releasing some molecules, drug molecules, and after some time it is releasing most of the drug particles. So this is in diffusion system and dissolution system, there are two diffusion control systems are there. One is reservoir system and matrix system. So this, if you call it as a reservoir, as we know that reservoir will be having water in between. Outside there will be concrete structure which will be able to hold the water in form condition. So in the same way here, this is the polymer matrix which is act as a concrete structure like that. So this polymer will be having a diffusion nature. So when it interact with the body fluids, then the body fluids enters into the polymer, and then that drug will be tried to come out through the diffusion process through the membrane. The same way, here in the diffusion system, where matrix diffusion system, we are making the carrier. We will be mixing both carrier and drug in a uniform. Wherever you see, this will be uniform throughout the structure. That drug and the polymer will be uniform. With the time, it will be releasing the drugs into the system. So after T2 second, most of the drug molecules will be diffused through the membrane. So here this polymer will be as it is, because here we are taking insoluble polymer. So the polymer structure will be as it is, and it will be excreted. And so this is as it is a monolithic. Wherever you see, it will be appearing in a uniform manner. So we can also call it as a monolithic diffusion control system. Suppose in dissolution control system, I am compressing the tablets by coating different coatings to the tablet. So here two layers, two coatings we have given. Here one coating we have. So when it is exposed to the body fluids, So polymers which is having a single coating will come fast, and then here no polymer is remaining here because it is dissolved in the body fluids. Because this polymer, whatever we are taking, is biocompatible and biodegradable. Hence, it is degraded and that drug released through this mechanism. Whereas in the case of a two-layer system, it will take time because this is having two layers, so it will take time. Initially, the first layer it will come out, and then second layer, second coated polymer will come, drug will come out. So this is called reservoir dissolution control system. So here polymer will not be there. So with time it will be degrade. Polymer also degrades, then drug will be released through the membrane. And when coming to the matrix dissolution system, whatever you told here, the same is applicable because here polymer matrix and drug is miscible with mixed and make a formulation. So with the time, that polymer gets degrade and then drug will be released. So with the drug releases and then polymer dissolves. In this case also, no polymer will be available. So both in either in the reservoir dissolution system or matrix dissolution system, polymer will be dissolved completely with the time because it is slowly soluble, so it will release the drug slowly. And the types of control release system and the preparation method we can discuss here. Diffusion control system, regarding reservoir diffusion control system is also called encapsulation method because here we are encapsulating the drug into the polymer matrix. So here core tablet is prepared and it is coated with the insoluble polymers. Whereas in case of matrix diffusion control system, tablet powder and polymer, insoluble polymer are mixed together and make into tablet. In the same way, dissolution control system, we call reservoir dissolution control system, drug granules are prepared and then they will be coated with the polymer, different concentrations of soluble polymers. Or it is also possible to coat different polymers depending upon the requirement also. You can coat first layer with a different polymer and the other layer with a different polymers. So concentration of slowly soluble polymers, because of this coating, it will be slowly soluble and then the drug will be released. And these granules will be packed into capsule in this case. Whereas in case of matrix dissolution systems, tablet powder and soluble polymer, slowly soluble polymer mixed together, and it will be mixed as and compressed into a tablet. And this is the types of control release system. Here, this is the drugs disc. It is incorporated in the polymer layer. So with the time it diffuses into the body fluids, depending upon, because here inside the matrix, the concentration of the drug will be more and outside will be less. So this drug will be diffuses into the body fluid. Whereas in this case, polymer will slowly dissolve and then it will release the drug into the system. Drug release is controlled by diffusion process in a diffusion control system. Whereas the polymers used in this case is insoluble and the release kinetics follows fixed law and perfect zero order reaction with throughout the releasing time, the constant drug concentration will be same. Whereas the dissolution system is a process because the drug solid particle gets converted into liquid phase and then it will be released. These polymers used in this system is soluble, slowly soluble polymer, and it follows the release kinetics as per Noyes-Whitney equation. Here no perfect zero order will be followed. And for one more example, I will explain you, because most of the people you might have seen this type of capsules, which will be having different types of granules. So there is a meaning for each color, because depending upon this coatings, this will be classified into different categories. Because the drug, as it is a drug release system, sustained drug release system, each coated color will be diffusing the drug into different timings. So here I am example, here I am taking diclofenac sodium as a drug and we have coated with the polymer and dumped into the capsule. So here yellow indicates uncoated, which is not just this is just diclofenac sodium crystals. So it will release the drugs because no layer is coated on that one. So it will release it in short period. Whereas if you come to orange granules, single coated polymer will release around 2 hours. So, and blue color granules will release the because this is a double coated polymer. So releases the drug in 4 hours. Whereas the red color is a triple coated polymer, and hence it releases the drug for 8 hours. Hence we can use this type of capsules as a sustained release systems. The same I explained in the dissolution system. Polymer membranes with the dissolution rates will be mixed. So this already explained. So in this type it will be working because of this reason it will be giving the drug for longer time. Hence we call it as controlled drug delivery system. And coming to the advantage of diffusion control system, a swelling control system can provide a sustained release over an continuous time, reducing the need for frequent doses and reduce the side effects because it is releasing the drug at a steady rate. The system will avoid the peak and drug concentration at any point of time. So that may cause side effects. So improved patient compliance. The following for the patient also it is easy because taking of tablets for 3 times in a day, this type of control system, if you take once in a day, it is sufficient and it will be simply, and that patient also will follow easily. And this simplicity schedules for non-invasive administration like oral and topical, etc. These also improve the adherence to the treatment. And this customization also can be followed by designing the control drug by fine-tuning the polymer type and degree of cross-linking. If the cross-linking of the polymer is more means, then the release through that system will be slow. Since you have to optimize the drug delivery cross-linking system according to your requirement. And it also can be optimized depending upon the environmental conditions like pH, temperature or ionic strength. And the limitations of diffusion control system, sometimes it may burst release may happen. Because of this experience, at a time the drug may be released, especially when the matrix swells rapidly at the start, initially. So complexity in formulation also is one more drawback, and to achieve the desired release rate, because you have to select the polymer and carefully, and then you have to make the formulations, which is very complex. So, and environmental sensitivity factors like pH, ionic strength, and the temperature also influences the swelling behavior of the polymer. Hence, we have to take the select the polymer accordingly where we have to apply, which part of the body we have to apply, because various body parts will be having various temperatures, pH, and accordingly we have to formulate the drug delivery system. And it also challenge maintain consistent and release rate in all conditions, because the affected body may be having different conditions and other part of the body will be having different conditions. So it is big task to maintain this type of challenges. And mechanism of controlled sustained swelling control release system. In this case, the polymer matrix absorbs water and swells, allowing the drug release to diffuse out. And the rate of the swelling is crucial to release the drug. So in this case, we will be selecting hydrophilic polymers, which are water soluble or water swellable polymers. The examples for hydrophilic polymers are hydroxypropyl methyl cellulose, carbapol, and polyethylene oxides, which absorbs water and gets a swelling, and then the release of drug may take place, and causing the matrix to swell out and expand. the matrix swells, the drug will release it through the swollen network. This, if the polymer, whatever we are using is a highly cross-linked means, then the drug, it is difficult to pass through that system. So accordingly, we have to define that cross-linking density, we call it as, we have to optimize that cross-linking density. So it can be controlled by polymer hydration also. The drug release rate is typically faster in high swelling matrix, whereas the drug diffuses more easily through this type of hydrated matrix. And the process can be controlled by adjusting the water content of the polymer's degree of cross-linking, as I explained earlier. And the examples for this type of is polyvinyl alcohol, hydroxy, propyl methyl cellulose, and chitosan are the good examples for this type of polymers which undergo swelling and provide control release, drug releases. And this is the swelling control release mechanism. So this polymer will be having will be outside, and inside the drug particles will be there. When it absorbs fluids from the body, it will swell it, and then here this type of drug it will be diffused from the polymer membrane. Mechanism, mixed matrix erosion control region. This is already discussed this thing. So bulk erosion and surface erosion are the two types available in the matrix erosion system. So this is the bulk erosion. So here polymer will be dispersed in the bulk polymer matrix, which will erode on the complete different parts of the polymer carrier. It will be released. So more drug molecules will be released. This is called bulk erosion. Whereas in the surface erosion, first drug will be dispersed in the surface eroding polymer matrix. So outside layers will come first. Then slowly inner parts of the system will come into the fluid. So this is called surface erosion system, because as the erosion is taking place from the surface of the polymer matrix. One more, another type of control release mechanism is osmotic controlism. This mechanism will be working based on the principle of osmotic pressure, from which water from the surrounding environment will be taken into the matrix and creating pressure that forces the drug to bring outside. So initially the higher water concentration to it works on the, suppose body fluid will be having higher concentration, whereas in the matrix, polymer matrix, with drug will be not having any water in that. So from high water concentration to lower concentration polymer membranes, it will diffuse through the system and then enter into the body fluid. And this follows the osmotic pressure gradient and zero order release system. Osmotic pressure gradient, when the polymer matrix in contact with an aqueous environment, the difference in the osmotic pressure between the inside of the matrix and outside the environment will cause water to enter the matrix, pushing the drug out through the orifice of the membrane. This will be explained in the with examples also in the next slides. Osmotic functions are well-known applications in osmotic control regulatory system, where the polymer membranes regulates the influx of water and release the drug through that mechanism. So it follows zero order release and the drug release at a constant over time, and this is especially useful in maintaining the steady drug levels in the bloodstream. This is the example mechanism we can explain. Suppose the membrane, so outside membrane, semi-membrane is there. This is the orifice from where the drugs particles will come out from there. This one. At time t, before operation of this one, it will be outside when we are making in the form of tablet. It will be like this. So when it is inserted, when you have taken with water, then it will come this phase. And from outside, the pressure will be pushed on that membrane, and then because of this pressure, it will be drug particles will be coming outside. Whereas in type B, which is called Ajia mini osmotic pump, this is also same thing, but additionally it will be having osmotic core without drug, whereas here osmotic core with the drug. So there will be, when applying pressure from outside, so this walls will be touched to this one forcefully, and then the drug releases through this system slowly. So this is the drug and the expanded osmotic, because this is of the due to the osmotic pressure from the outside. So it will be pressing this system and the drug will be released out. So this is only for water soluble drugs and not for highly soluble or insoluble drugs. This mechanism. Whereas this type of osmotic pumps will be used in case of implanted tissues. Force-based osmotic pump will be using. And coming to the example of ion exchange control release system. So for example, I am telling here, you and your friend are discussing together, and in between your friend has come. So you will be discussing with your friend and your friend will be keeping idea. In the same way, the system also, depending upon the ion exchange in this system, I will be explaining this one. And suppose if this is one, R is resonate here, A is anion, and D is a drug. Here this is in vivo. It is when you are making the tablet or in the packet, we are describing this one. So the drug particle is along with resin is called resonate. So when it is inserted into the body, so as I told you earlier, U is a resin. When in your best friend is place. So when drug matrices along with the drug comes into this one, into the stomach. This is the stomach environment. So if it is higher acidity is patient is facing, this higher number of H plus ions will be exchanged with drug, and then it will release the drug, the system. So D is having more affinity towards R. Hence, H plus ion will be having more affinity. So it will release the drug, and then this is in vivo. This is in vivo. This state status is in vivo system and then it will come out of the body. And the ionic drugs binds to the resin through electrostatic. These ionic drugs will be binding with the body through electrostatic interaction and released by exchanging the similar charged ions. So H plus and D plus are similar charged ions. So it will interact and then release into the environment. So release rate can be controlled by factors like pH. Like suppose if H plus ions acidity is more, means it will release more drug. So with this idea, we can use this principle to release the drugs using for acidity, like labiprazole. This is also called as intelligent drug delivery system, because it will depend upon the, suppose if the patient is having a higher acidity, the higher H plus and Cl drug will be released. So it will be controlled within no time. And greater the exchange ions will be taking place. So complexation of formulation depends upon the ionic exchange capacity, particle size of the resin, size of a drug and ion, and drug solubility, pH and ionic strength. It will be depending. These complexations will be depending on these things. Whereas drug release and absorption depends upon the ionic strength and pH. Cross-linking of the resin. How much this resin is cross-linked? Depending upon that, it will be released. Here in this case, drug solubility also will affect the release rates, and membrane permeability at the site will be possible. And this will be excreted through the system after coming out after releasing the complete releasing of the drug. Very minute quantity may be there, which is not soluble. So it may come through this excretion. And this is because this is coming about biodegradation and biocompatibility, and the site of administration, it will be depending. So ion exchange rate in control release system have used can be used to deliver the ionic drugs only. The systems are used only ionic drugs, which is having charges. So they are not suitable to deliver the non-ionic drugs. They use resins, which is basic bone of the in this system. Resin is the basic bone of the carrier, and resin must be water insoluble, because if it is soluble, then it will be burst release may be there. Because of that resin, immediately or with time, with a small amount of time, it will dissolve, means it will be immediately release the drug, and it will be leading to the side effects. Hence, you have to consider in this case water insoluble polymers, which will be biocompatible polymers. It will be there, but it will not be having any toxicity in the system body. And the resin must be cross-linked with the abundant ionizable functional groups, because in order to release the ions with when it comes to the stomach or any other system, it should be having some ionizable functional groups. And the common widely used examples for backbone resins are, this is divinyl benzene, cross-linked polystyrene, and polymetacrylate polymers are all widely used in this type of control release systems. Ion exchange is a reversible process. Ions can be exchanged between liquid and the solid resin. If suppose molecular weight and the water insolubility is directly proportional, they are not absorbed by the body and will be inert. And the classification of resins you can see here. So cation exchange resins for basic drugs you can use with anionic drugs. So in case of anion exchange resins for acidic drugs, we will be using cationic drugs. So here strong acid cation is sulfonic acid, and functional groups attached to a system, and divinyl. This divinyl benzene is a common polymer in these cases. Here strong acid cation will be used. In this case, weak acid cation will be used, and that is carboxylic acid functional groups. Whereas strong base anion will be used, that is trimethyl ammonium chloride attached to the styrene and divinyl polymer. And in case of weak basic and anion, and ammonium chloride is used as a for functioning the resin, like styrene and divinyl benzene. Examples for this type of this Dovex 50. Example for weak acid cation and carboxylic acid, this is amberlite. This Dovex I and amberlite IIR 4B. Ion exchange resins are also used in making the mask for the syrups or other materials which will be not interested by the children. Suppose if the smell or taste is different, the children will not take. So in order to avoid that one, this type of ion exchange resins can be coated on the as a masking agent to that delivery systems, which reduces the bitter taste of some drugs. As the drugs are chemicals, they may be having some different taste of the different drugs. So in order to that, we will be masking with this type of ion exchange resins. And the polymers used for this purposes typically are polymers coated with acrylics, cellulose acetate phthalate, ethyl cellulose colloidal dispersion, ethyl cellulose wax, gelatin-based polymer complexes, and hydroxypropyl methyl cellulose. And depending upon the shape of the drug molecule, the release system will be varied. So diffusivity, ability of a drug molecule to pass through the membrane is called a diffusivity. It can be denoted as D. D is a function of molecular size. If size of the molecule is less, then it easily diffuses through the membrane, and it is a good candidate for use in controlled drug delivery systems. If the size is more or complex, then it is difficult to diffuse through polymeric membrane, and it is a bad candidate for controlled drug delivery system. So if it is spherical system, molecular weight up to less than 150 will be good candidate, whereas the chain molecules will be up to 400, less than or 400 can be a good candidate for using in this ionic exchange reaction mechanism. The solubility, poor water soluble drugs are not suitable for this type of controlled delivery system. And average water drug soluble are good and suitable for this type of systems. And the good water soluble drug are good and suitable candidate for controlled drug delivery system. Suppose if the higher the water solubility, better the drug release will be there, and rapid increment in plasma drug concentration will be there. Hence, pH dependent solubility also creates problems in formulating this type of formulations. BCSS class 1 and class 2 are good candidates, and class 3 and 4 are suitable candidates. So non-ionized forms are better absorbent than ionized forms because ions will be there, and in the fluids also ions will be there. So because of exchanging of this ion, effectiveness will be better. So regarding the drug stability, what type of stability should be there in order to select the drug for a controlled delivery system is, it should be stable both acid, base, or in gastrointestinal fluids are the good candidates suitable for this type of drug delivery systems. Drugs which are stable even in enzymatic degradation are good candidates for this type of DDS. If drug degrades in stomach, small intestine, and are not suitable for this type of candidates, because this ionic exchange will be taking place normally in the stomach itself. So if the drug degrades it there itself, it will not function, and whatever the purpose will not be solved. So unsuitable candidates are very difficult to formulate into the prolonged release systems. Several modified release methods have to adopt to make the unsuitable candidates to make them suitable for CDDS. And the rate of absorption also will depend on the rate of release. So whatever the drugs we are using, it should have high absorption rate constant, then it will absorb rapidly. And they are good candidates for controlled drug delivery system. Drugs with low absorption rate is equal to slow absorption, and hence these are not suitable for controlled drug delivery systems. And coming to the stimuli responsive system, there are different stimuli responsives of the controlled drug delivery system. This is also called as smart release because it releases the drug with effect from varying parameters like temperature, ionic, and enzymatic. So pH responsive system, examples for pH responsive system are polyacrylic acid and chitosan will be swell and degrade in response to pH changes. So this type of polymers will be used in fabricating the drug delivery systems for pH responsive systems, such that occurs in different parts of the gastrointestinal, because it can be used in gastrointestinal tract and allow them for site specific drug release. Hence, this type of wherever pH is involving, then if you select these polymers and the drug will be released as per the pH present in that gastrointestinal tract. And the coming to the temperature response system, polymers such as polynisopropyl acrylamide undergoes phase transition in response to temperature. With change in temperature, this type of polymers will change its phase. So hence, this polymer are candidate for temperature responsive sensitive drug delivery system, which releases the drug at whatever the specific temperature we want to release, because whatever the affected body in the system will show different temperature compared to other parts of the body. Hence, in these cases, these are very good candidates to use in the drug delivery system. And another type of responsive system is enzymatic response system. This will designed to degrade the presence of specific enzymes. These polymers undergo degradation in the specific enzymes condition. So it can be used for localized drug release in the areas where these enzymes are present, like such as digestive enzymes, enzymes at tumor or sites, etc. So these are the examples for the stimuli responsive system. Suppose there are some other responsive system also there, light responsive system, redox response system and magnetic response system. This is the smart stimuli response drug delivery based on the cyclodex. And this is classification. The license is the pH value and enzymatic and temperature type of response system for this purpose. And these are different, can be classified as a physical stimuli response system, biochemical stimuli response system, chemical stimuli response. Under chemical stimuli, you can classify as redox, gas, and ion system. pH also it will come under chemical. Whereas in biological, enzyme, protein, DNA, and glucose type of this stage is called biochemical response system. For this environment, that enzyme polymers, whatever we are using, it will be responding accordingly, that will release the drugs. Whereas physical stimuli, like light, temperature, magnetic field, etc., all comes under physical stimuli system. So here are the examples for pH temperature stimuli response release and pH stimuli release. Suppose this is the drug matrix along with the polymer matrix. In the presence of here, this is tumor tissue, whereas this is normal tissue. Red color is a tumor tissue. So in which temperature will be slightly varying compared to the normal tissues. So it will be targeting this area and release the drugs at that particular tumor phase. So in this phase, suppose this polymer, when it goes under change into the body, where pH changes compared to normal, then it will undergo swelling and then deswelling of the polymer takes place. An ionic bond of the polymer will take place and then release the drug at as per the changes in the rate. Suppose once it changes it, once it is released into the pH system, once it is balanced, the pH is balanced with the other one. So then it is cured. So hence it will not be consumed anymore. Then it will be extracted. These are the some of the examples for temperature response system: polyanised propylchromide, propyl methacrylamide, poly N-caprolac cyclopolyacylamide, which will be having LCSTs as follows. And this is the applications of pH response system hydrogels. It can be used in oral drug delivery system and also can be used in injectable drug delivery system and also in transdermal drug delivery system. And the application of this type of pH sensitive response system is to treat the diseases in gastrointestinal and intestine and colon target delivery also can be used. Protein and peptide drug delivery systems can also be used. Transdermal patches can also be used, and hence it is also used in cancer targeting drug delivery applications also it can be used. And these are the pH responsive polymers. It will be classified into two: natural polymers and synthetic polymers. Synthetic polymers are alginate, cetogen, polon, carboxymethyl cellulose, hyaluronic acid, starch, dextran, polyurethane, etc. Whereas in synthetic compounds, this again classified depending upon the composition and the structure, it can be classified. So under composition based polyacids, polybases, polymers with acid and base label linkages will be used. So this type in the structural based linear block copolymers, star copolymers, star polymers, brush and comb polymers and hydrogels will be used in pH responsive polymer system. Physical or mechanical disruption, it is also called as burst release, because this is physical. At a time it will be released depending upon the requirement. So burst release occurs when the drug is rapidly released from the matrix. This may be happen due to the change in the matrix structure because or physical disruption of the polymer. So polymer disintegration is some polymer matrix physically break apart. Example, by disintegration in water or change in pH. Suppose some of the polymers are sensitive to pH. So when it is passed through that pH, higher pH or lower pH, it may act and disintegrate that. In some cases, even in water also, it will get under disintegration and resulting in the rapid release of the drug, which is sometimes used for initial loading. So suppose if any person is having a severe stomachache or such type of things, these burst cases will be, burst release systems will be used. And microsphere rupture. In this type of systems, micro or nanoparticles can be used in combination with the polymer matrix, which ruptures or collapse, leading to the rapid release of the encapsulated drugs. Microspheres, liposomes, and nanospheres can exhibit a burst release under the certain condition. So the drug release mechanism through polymer are highly varied and can be selected and as per the engineered to meet the specific therapeutic requirements of a drug or treatment. Whether it is should follow through diffusion or swelling or erosion or osmotic pressure or ionic exchange or stimuli responsiveness, we can choose this and accordingly you can formulate the formulations. These mechanisms allows for the creation of highly tailored drug delivery system that provide control, sustain and targeted drug release. And this is the important topic, that targeted drug delivery system. Actually, we are giving injections and it is going throughout the body. So it will not be effective that much compared to target. Hence, with this concept, they have targeted the targeted delivery system where exactly the problem is there. We are giving this. This indicates this target. This is our target. So when the whatever medicines we are giving, it has to target the specific point where it is infection or some deficiency. This one problem is there. So here we have given injection to a person, and in a conventional drug delivery system, whatever the drug injection we have given, it will pass throughout the body. So here intensity will be less. So this is the problem actually. But very less amount is coming to the system. Hence it will take long time to cure. Whereas in this case, this is targeted delivery. We are giving the injection which is have to work in a particular area. Lungs only it has to work. So it will not go to the other parts of the body and it will not affect the healthy tissues also. So once it is go to the target, say it will not take much time to cure the system. So this will be helpful. This type of delivery system like targeted drug delivery will be helpful in these cases. So here the concept of this is this is the other tissues, whereas this is the targeted. We have targeted this area. So when it is passing this one, it is not touching other tissues. You need only passing because of this nature, receptors nature. We have targeted and accordingly we have formulated and we have given the medicine to the patient and it is going to target the tissue where we have targeted. So hence it is a drug availability and accumulation will be more here and the treatment will be fast also. So comparison, when we compare with the conventional and targeted drug delivery system, conventional drug delivery system is distributed throughout the body. So more volume of distribution will be there and less effectiveness will be there. Whereas in targeted drug delivery system, drug is concentrated only at the specific site. Hence, less volume of distribution will be there and effectiveness will be more. So more in case of conventional, more dose but less drug at the site of action will be available. Whereas we can reduce the dose here. Because of increasing of the dose also, there may be having side effects. In order to avoid these things, we have chosen this targeted drug delivery system. So here less dose is sufficient, but effectiveness will be more at the site. Comparatively less therapeutic action in this case, where in conventional drug delivery system, whereas here in the targeted drug delivery system, more therapeutic action will be noticed. Here in the case of conventional drug delivery system, healthy tissues also may be unnecessarily exposed to the drug and leads to the side effects. So whatever the tablet we take, there may be certain side effects. So we have to take care when we are taking this type of medicines. And a healthy tissue will not be exposed in this case, where in case of targeted drug delivery system. So it will be very useful. And why it is required, targeted drug delivery system, why it is required. Actually, as per I discussed earlier, in normal conventional drug delivery systems, in regarding the pharmacokinetics, low half-life will be there and a high volume of distribution will be there. Regarding the pharmacodynamics, low specificity and low therapeutic index will be there. So in case of pharmaceutical, low solubility of the drug will be there and low stability also will be there. And in case of pharmacotherapeutic, high dose is required and adverse effects will be there, and patient cannot be complaints in this case because he has to take a lot of frequency will be more in this case. So it will not be that much complaints to the patient. So in order to avoid these facilities, we have achieved this targeted drug delivery. This concept has come. So this is the definition. It is the targeted drug delivery is defined as the drug which is delivered to a specific location or specific site rather than the whole body. So drug concentration in the tissue of interest will be targeting at the site. So wherever it is required, which tissue it is required actually, so there only it will be targeted. So also it is also aimed to high efficacy at the site of action and no toxicity to healthy tissues. And it is a combination of diverse field. This is so who are working in this targeted delivery system will be diversification fields may be involved in this type of formulations like polymer science, polymer science expert is required to make the formulations to make the carriers which is suitable for that particular target tissue. And pharmacology is required to make the suitable drug for that particular targeted site and suitable for that polymer. And bioconjugate chemistry is also required, molecular biology, because these people only will be knowing about the organic system in the how it will affect and how it will perform. These things will be known by these people. So combination of these all fields will be healthy to make the targeted drug delivery system. So if you see this picture here, this is only eye. It is targeting eye. So we have doctors will suppose if any patient suffering with eye problem, when he goes to doctor, he will particularly work with that particular site only. He will treat this system only. In the same way, pharmacist also, pharmacologist people also, they have based on this concept, they have found this targeted drug delivery system. Why doctors are using only, earlier days they used to give anesthesia throughout the body when if your patient is undergoing surgery. However, after this local anesthesia comes, that also one of the concept in taking this targeted drug delivery system. Based on this, doctors have come successful in selecting this targeted site and treating that same site. So why can't we work for this purpose? So with this concept only, this has come, this targeted drug delivery system. So they have formulated this type of these systems to work at the specific site. These are the type of various type of carriers that are used in the targeted drug delivery system. Maybe they may be 0-dimensional, 1-dimensional, 2-dimensional, mesophorus, liposomes, micelles, dendrimers, and other things. So this is 0-dimensional. This is fullerenes, and these are the 1-dimensional nanotubes, and the graphene are 2-dimensional, and these graphites are 3-dimensional. Carriers are required to carry the drug to the target system. And these are the different approaches for targeted drug delivery. This is based, it is classified based on the modification of the drug particle, based on the targeted nature, based on the area of target, based on the site of action and the nature of disease. So this targeted deliveries will be defining by modification of drug particle, where it has to work and how to modify this drug system. And depending upon the nature of the target, which type of, whether it is interior to the system or exterior or single target is required or multiple targets are required, depending upon this it is classified. So target delivery systems are classified in this way: active targeting, passive targeting, biological targeting, physical targeting, chemical targeting. These are the various different categories depending upon the requirement. So the benefits of the target delivery system is it can be worked in the local area, wherever you want, and it will release the drug in the controlled way and reduce the side effects. Efficacy will be more, and toxicity can be reduced and reduce the dosage also. These are the applications of benefits of the targeted drug delivery system. So mechanisms of targeted drug delivery system, there are different targeting systems like passive targeting, active targeting, physical targeting, double targeting, dual targeting, and stimuli responsive targeting. Now one by one we will be discussing. This is the passive targeting. In this case, drugged particle surface is not modified at all. Only will try to reach the target area because the body's nature responds to diseases, inflammation, or infection also. Example, enhanced permeability to endothelial cells in the area of cancer cells, like endothelial cells near the cancer cells have EPR property. EPR is enhanced permeability and retention. If you take this example, this is normal cells, whereas this is the cells which have suffering from cancer. So when we inject this target delivery system into this system, so normally endothelial cells, this cancer cell will be loosened. So whatever the drug we give, it will pass through this system only. So it will not, because this is tightened, because it is healthy tissues. So it will not, this endothelial cells will not be loosened. So the drug will not be passed through this system. So it will pass through this loosely endothelial cells only, because this is the targeted area. So the drug will reach to this target only and it work. So a healthy tissue it is not reaching. So it will be good for to reduce the side effects. It follows the target to systematic circulation. It can be used for anti-cancer agent to treat the cell cancer area, affected area. Then this is the active targeting. Suppose this is the temperature sensitive liposome molecules, and this is the drug with anti-cancer related to the anti-drug. So normal healthy environment will be having the temperature around 37 degrees. Whereas the area which is suffering, having a cancer cell will be having this type of this structure, which will be loosely structure will be there because of this temperature variation. So it loses the structure. So hence the drug, whatever you injected, it will pass through this cancer affected area cells only. Here we are giving because this temperature variation, because of this temperature only, because temperature will not be that much, but even 2-3 degrees also will vary. So it will affect accordingly. In this case, temperature sensitive polymers can be used for this targeted delivery to carry the drug. So active targeting is drug particle is placed in a carrier, and the carrier will be modified as per the microenvironmental or target cells. Reaches drug will be released at the target site. Examples are heat sensitive polymers, and examples another example is coating of surface with ligands like metal ligands. Suppose if you are passing coating with iron ligands, and also it can be coated with biological ligands like proteins, polysaccharides, and also peptides, tissues, albumin, bioadhesive molecules, etc. And the ligands that is designed based on the receptors present in the target. Suppose if having any issue in that particular area, there will be receptors. Which lesions will be accepted this reason accordingly. The lesions will be ligands will be selected, and there are 4 orders of active targeting. This first order, this is not the release way of first order or second order. This is different, and depending upon the site, the first order capillary, this targeting will work only on the capillary blood or targeted side. Whereas in the case of second order, specific cell where the tumor cells is available, there only it will attack and then it will release the drugs. So in this case, intracellular components, it will work third order reaction. Whereas in case of fourth order, it will undergo, it will also works in the place of DNA, proteins, like that. Like these are macromolecules. So in such cases also, this type of active targeting is possible. This is the active targeting that I have explained. So here there are different types of ligands. These are some of the biological ligands like proteins, polysaccharides, peptides, aptamers, like that. So this type of these structures, this is called a drug. This is a drug, whereas a injection into the body, this will be injection into the body. This is the drug, whereas it will be hold with ligands, whether it is biological ligands or other ligands. It will, when it is combined with this one, so it will accordingly, it will give active targeting. Those receptors will accept this type of ligands. Accordingly, these formulations can be modified. The physical targeting. Physical means that are used to alternate some of the physical characteristics of the target site. Example, using the magnetic field or electric field or ultrasound like system, we can be used in this physical target. So suppose we are giving injection which is having tumor. This is here tumor cell is available. So we are putting that magnetic field here. So and we are injecting the iron ligands, drugs with iron ligands. So it will not go anywhere into the body because it is attracted towards the magnet. So this iron particles will go through the to this site only, and it will function as a targeting drug delivery system. In the same way, this ultrasound also can be used wherever the ultrasound is variation is this normal system. It will pass throughout this one. But this polymer are prone to ultrasound. So when it reaches to the area where it is ultrasound, it is applied, it will break that polymer matrix membrane will break and then will release the drug. The same way, electric field also you can apply. This magnetic field already applied. In the same way, which will be prone to light. Suppose if you are putting this light, this type of ligands will go to that target only, and there it will release the drug. So this is dual targeting. Here in this case, we are targeting some sensitive parts will be there. So in that case, we have to use this dual targeting. Here targets will be two to reach the target site. Initial target will be one different, the actual target will be different one. So in order to overcome this one, we are following this dual targeting. Here carrier molecules are functional with the two targeting ligands. See, if you see these colors, two ligands are there here, blue and pink. So when it is a brain tumor different from peripheral tumors, drug delivery to brain tumor is largely restricted by the blood-brain barrier. So here it will be not allowing to pass through this one. So to pass through this one, different type of ligands will be linked to the polymer carrier or whatever the carrier we use for the treating that specific target. So we will be attaching two types of targets. So here target one will be we have to cross this blood-brain barrier, and then it has to go to specific target. This is infected area where brain tumor is available. So it has to go to this part. So the development of dual targeting delivery system is still in its early stage. So if you work on this, people who are working in this area may be having a good future, because it will be helpful for this is very costly treatment also. So lot of chances will be there. So I suggest people who are interested, they can go for this type of dual targeting. And this is double targeting. Double targeting is different from dual targeting. Here we are targeting two targets. One target is to reach the specific organ or tissue where we have to get treated. And another is rate of drug delivery to the target site. In previous case, we are crossing two targets, but whereas here we are targeting the specific organs plus the control drug release system is applying. Hence, it is a double targeting. We are using two targets here. One target is specific organs, and another one is rate of release of drug at the targeted site. And the advantage and disadvantage of these things is it is less dot and high efficacy will be there in this type of things. But disadvantage is chances of immune system reactions on carriers will be due to the target. These are less side effects will be there and reduced toxicity will be there, and needs very high sophisticated technology is required, because as it is early stage, with the time maybe this can be overcome, because a lot of people will be working and new ideas will be there. So we can achieve this target. But as of now, it is little early stage. So sophisticated technology we have to develop to meet this requirement. And avoid first pass metabolism, one of the advantage, and a high manufacturing cost and time. This also can be reduced with the time, because usage is more automatically the cost of the systems will be decreased, and the technology preparation technology changes also, it will reduce the cost. So drug remains at the tissue for a longer time, and it will treat till the drug is available, and it will not be available in other sites. So it will be working with the same this one. And so that is one of the advantages of this system. And it is difficult to maintain stability to dosage forms released erythrocytes, like 3 or 4 degrees variation may not be much. Maybe some of the things may not be response for this small temperature variation. So we have to work in this area also. And whereas in case of infections, it is the drug will reach only to the microorganisms and not to all the human body. And it is in this the disadvantage is difficulty to predict and fix the dose at that region. Role of the polymer matrices in the target, as actually I have already explained the functionalization of the target ligands. All these things I have explained, and drug encapsulation efficiency. These are drug encapsulation in polymer matrix also explained. Role of polymer matrices targeted this all type of pH response. All these things I have explained. These things. Advantages also I have explained. And where can you use this targeted diagnosis in treating that cancer therapy, like polymeric materials we can use, and this can be functionalized with the cancer cell, and specific ligands can be functionalized, and then we can target the where you have to actually cancer cells. It will be specifically this type of ligands we can use to target that cancer cells. So, and expressing the receptors will be there at the target. So that should be according to that receptors, we have to modify the ligands functionalization. And these can be encapsulated chemotherapeutic drugs like doxorubicin or paclitaxel and etc. Drug will be delivered directly to the tumor, hence reduce effects on the other healthy tissues. In this case, polymeric micelles made of polyethylene glycol and polylactic glycolic acid can deliver this type of doxorubicin to the tumor cells. So these are the polymers can use target in cancer therapy for in case of targeted drug delivery system. So in case of inflammatory disease treatment also, this type of targeted delivery system can be used. In this case, we can use as a polymer hydrogels for anti-inflammatory drugs or biological agents to the inflamed tissues. The design and the release of the drug in the response to local inflammatory markers like elevated temperatures or enzyme activity or pH variation, and hence the therapeutic effects will be increased and reduce the side effects. Chitosan hydrogels we can use as a targeted treatment of rheumatoid arthritis and deliver the drugs directly to the inflammatory agents and reduces the exposure, systemic exposure and gastrointestinal side effects. Suppose normally we are using arthritis tablets, which also leads to the gastro problems also. So in order to avoid these things, this type of chitosan based hydrogels we can use, and then it can be functionalized with targeted ligands, and then we can overcome these type of problems. And it is also useful in central nervous system drug delivery, which is a very sensitive part. So as I explained, it has to cross blood-brain barrier. We have to use two types of double targeting system we can use in this area. In this case, we can use central polymeric material functionalized with transferrin or other blood barrier targeting ligands, which can be used to cross the blood-brain barriers and deliver the neuroactive drugs or gene therapy directives also can be directly to the brain. Instead of passing through other body system, it can directly attack the affected brain part. Polyethylene glycolated nanoparticles are loaded with dopamine and can be used as a targeted delivery system to brain region in Parkinson's Parkinson's diseases, or enhance the drug delivery to reduce the side effects. Now, it is also can be useful in vaccine delivery, and polymeric materials like microspheres can encapsulate the antigens, and then it can be released in the controlled manner to immunize the system and improve the vaccine efficiency for a longer time, and the immunization responses will be better. And this PLA, polylactic glycolic acid nanoparticles, designed to deliver the antigens for malaria vaccine or mRNA vaccines and protect the antigen from degradation. When normal vaccination, if you give, there is a chances of degradation. Whereas this type of targeted delivery systems, if you modify the polymer matrix with this type of ligands, and then you can enhance the immune system recognition also, and other degradation of antigens can be taken place. And the challenges and future directions of targeted delivery is tissue specificity of tissue where we are targeting the cell. So polymer based system can achieve the targeted delivery because it can modify as per the requirement by functionalizing with related ligands, and ensuring the sufficient specificity for particular tissues is a challenging target, and hence strategies like multiple ligand conjugations, like I told earlier, two ligands conjugation we have used. So multiple ligands conjugation also you can implement for this type of tailoring this type of surface properties and being explored to enhance targeting accuracy. And scalability and manufacturing this type of production of this type of nanoparticles and polymer matrices is meet a regulatory standard for clinical usage, and which will be complex and will be costly also. And so hence advances in the manufacturing technologies such as 3D printing or microfluidic devices also will be plays key role. Once it is developed, this type of activity targeted delivery may have promise, and improve scalability also chances will be there. And immunogenicity, like some other polymers can be engineered for biocompatibility, which is having some other materials may still provoke immune responses. Depending upon the immune responses, we have to develop this type of polymers also for using in this type of delivery system. And a lot of research is going on this usage of these natural polymers for this type of immuno rejection areas. So next topic is ocular drug delivery system. Here first we are discussing about salt gel system. Here actually salt means solution, whereas gel means it will be in the form of a gel. So which is not a hydrophobic, which is not hydrophilic. So here in this case, we are dropping from this we are putting the drop into the eye, because this eye will be having different pH values, different temperatures, and will be there, and there may be some ion activation may be possibilities. When you drop this drops into the eye, Because of this present in the environmental condition in the eye, it forms gel. So which will help in the, suppose if normal drops means it will not be run away, it will run away within no time. So effectiveness will be less. So when this type of gel, salt gel system use, this cell gel when it and this drop enters into the eye, it will form into the gel. So there is a chances of increasing the bioavailability. Hence there will be lot of more time will be there compared to liquids. So treatment efficiency will be better compared to normal this type of eye drops and ointments. So eye drops, these are the ocular systems we are using nowadays. Eye drops, eye ointments, eye gels, and currently we are discussing about this one, which is very important in the future. So eye suspensions also, and this is one of the important this AccuSets, one of the important devices to treat the eyes. So these are the drops we are using. So when we are, these are the sterile isotonic liquid preparation meant to instill into the eye. When you are injecting, that should be very sterile and isotonic because it is sensitive part. If you, without steriling this type of drops, if you put inside, it may affect the, and there may be chances of damaging the eye and forming infection also. So liquid preparation, low viscous, high drainage will be there because of this normal systems. So not preferable nowadays. Hence nowadays doctors are advising this type of, is not advising for this type of, and so nowadays they are using this type of gel system only. So limited ocular absorption and only less than 5% of drug delivery drug will be absorbed through this type of drops. So if it is gel, it will be more time will be exposed to the eye, and then the effectiveness and the absorption rate of the drug will be more. Hence treatment will be effective. And the preparation of this will be, all the ingredients are completely dispersed in a solution, and it should be isotonicity must be adjusted by class 1, class 2 methods. And this is, I am not going to the deep. And the eye ointments comprises of sterile, semi-solid and anhydrous preparations meant for illustrating into the eye. Drug is dispersed in suitable non-irritant bases like mineral oil will be used in this type of formulations. So it will be popular for pediatrics. Children's it is using most of the this type of things. And it also provides longer contact time compared to conventional eye drops and increased bioavailability. And the limitations with this is interferes with vision. Suppose when you use this type of things, it may affect the vision also. And because of its greasy nature, blurriness will be there. So the type of complications will be there. So maybe with this time, this type of things can be avoided by developing the technology. So here what my example, earlier example what I told, this drops when it is injected, it forms a cross-linking with the fluids present in the gel. So it will be exposed whether it is a temperature variation, whether it, the ionic changes will be there. Because of this, it forms this polymer. This also gel also will be having polymers like carboxymethyl cellulose polymer will be there. So this will undergo cross-linking and it will form gels. Solution remains as a solution in a drop bottle. When it remains gel on the contact with eye, it forms gel in presence of temperature and other pH, other conditions in the eye. So this gel will be having high viscosity, hence drainage will not be there much and more contact time, best availability. So it can be used as a prolonged release of drug and best availability devices. So more preferable than normal delivery system. This is one of the recent trends. It is going on AccuSets. This is also known as ocular inserts. This is we are inserting into the eye. So hence we call it as a ocular inserts. These are prepared by sterile preparation, multi-layered, solid or semi-solid devices placed in cooled sac, means conjunctal sac. and the size and the shape of designed as per the requirement of by ophthalmic application. And it is a type of drug dosage form. And this also will be having drug. It can be used for both vision and as well as treating any problems is there in the eye. And the drug will be released in a constant rate by diffusion mechanism. So the corneal contact time prolongs duration of action and improving availability will reduce the frequency of it. So normal drops will be using, suppose if whenever you go to doctor, eye drops, he will suggest to put at least 3 times in a day. Because of this running time, it will run away as soon as we put it into the eye. So effectiveness will be there less. Hence, this type of accusers can be used to reduce the frequency of administration and achieve the. It will be also convenient for the patient to follow, because instead of taking the medicine for 3 days in a day, so if you put this accusers, it can be stayed for 2-3 days as long as it is required. We can keep it and then we can remove it off. So example for this type of accusers is, it is already available in the market in the name of pilocarpine accusate. This is the first product developed by Alja Incorporation in US. This is the cul-de-sac. We call it as cul-de-sac. So we are putting this pouch type structure, which will be having. It will be keeping here, and then it will be working in the eye. The lacriminal fluids is secreted and opens interiorly between the eyelids also. So this is the system, bulbar conjugates and palpebral area. So it will go to this area and it will work. And these are the 3 components present in the accusers. So first one is a transparent rate control membrane, and the second one is annular ring, which will hold this one. This polymer, it will be hold in between the second one, and the third one is drug reservoir, where the drug will be encapsulated in this layer, and the transparent rate controlling membrane is both. So outside and top and bottom will be transparent rate controlling membrane will be there, and the drug will be released into the reservoir in a controlled manner. This is the structure of this accusers. And they are primarily using for vision correction normally. But nowadays this concept has gone to using drug delivery devices also. So these accusers will be dipped into the drug solution and will make contact lenses, and it will be used and soaked in the drug solution. So it will be used as a device for vision correction, and as well as a controlled drug delivery system also it can be used, this type of accusers. So this will be prepared by hydrophobic polymers. Normally hydrophilic polymers are widely used in this type of accusers manufacturing. So polymers cross-linked covalently to form 3-dimensional structure. That is, whatever I told is cross-linking or hydrogels are a 3-dimensional structure will be there. When it is injected into the eye, that polymer will undergoes cross-linking and then it will form gel. So this contact lens nowadays pre-soaked in respect to drug solution, and it will be used for treating the eye and as well as vision correction also. It will act as a dual function. So this is particularly useful in the case of astronauts. Suppose if they go into the space, these drops will not fall down. So it is difficult for them to put drops into the eyes. So with this intention, Russian people have developed this type of accusers for this cosmonauts. So the drops is not due to the gravitational force, the drop cannot be injected into the eye. So they are using this type of films made from acrylamide. This is the one accusate that can be used in the space. And these are the mini discs. These are profiled convex outside and concave inside. And This is the one which can be used in the eyes, which like similar to that of contact lenses. And this dosage form is either hydrophilic or hydrophobic form will be there. It will enable the extended time period of release of water soluble and purely soluble drugs. This type of discs can be used within the eye for treating the. So collagen shield protects corona and cornea and RX dry eyes. So this is abrasion area where this is collagen shield. And so this type of collagen shield can be used as a contact lenses plus collagen shield. In the both ways it can be used. This is bioerodible and degrades an eye itself and dissolved in liquid and lubricate the eye also. This is a natural coating. This collagen shield is a natural coating that is when combined with this abrasion, this type of disc, it will be compatible and transparent and safe also. These are using in for fabrication of IR to RX dry eyes. Those who are suffering from dry eyes, this type of this will be used to for natural artificial tears. So when we can deliver the drugs also along with the lubrication also, it will be happening. So hydrate the drug solution before placing into the eye, and then it will be having dual action. So some of the disadvantages with regard to the accusers are, it will be discomfort for the eye movement. Suppose if you are moving the eyes, it will be discomfort when it is injected into the eye. And interfere with vision also. It will be blurry and it will not be easily not able to see properly. And visual disturbances also will be there. So and loss of device also during sleeping or while washing or rubbing the eyes also take place. So these are the disadvantages of this. So regarding the advantages of this accusers, can increase the contact time, availability, bioavailability and ocular permeation, and better therapy and efficacy. So frequency of administration can be reduced and systemic absorption of a drug will be possible, can be reduced, and the adverse effects also will be less. The patient complaints will be more, and the possibility of drug targeting is possible with this type of devices, and possibility for controlled drug release also possible. And the type of polymers that can be used in ocular devices is polymer, natural polymer, chitosan, alginate, gelatin, synthetic polymers like polylactic glycolic acid, polycaprolactone, polyethylene glycol, and other eudograte. These are the methacrylatic copolymers which are sensitive to pH and temperature. And it is also releases the sustained release formulation, which is attractive coatings and a masking agent. This eudograde methacrylic polymers, when you inject into the this one, it helps the as a masking agent, like smell it can reduce and it will not take taste and protect the actual active pharmaceutical ingredients. Suppose if you use this type of system, then it can emit the degradation, because this active pharmaceutical ingredients may degrade while transferring from one place to the site. So to avoid these things, this type of polymer accusers can be used. Example of other ocular inserts and films like axillary accusers and contact lens drug delivery devices also it can be used. And these implants are biodegradable, like can be used polycaprolactone and polylactic acid for long-term delivery of a drug through dexamethasone. This already discussed. Now we will go to mucoadhesive conventions. Normally, this is the conventional formulation for mucoadhesive, and this is the mucoadhesive. When we are taking the drug by normal conventional formulation, as soon as it goes to the stomach, and it will go to immediately release the drug, and then it will be excreted. Whereas in the case of mucoadhesive, muco means actually wherever it is the body, it is having wet condition, this type of polymer matrices will hold firmly to that specific site. Here, this type of mucoadhesive formulations, we can use drug plus mucoadhesive polymer will be there, and it will adhere to that specific area where it is actually has to release the drug. Then we can achieve the drug release, targeted drug delivery plus control release system. So because this vehicle I have mentioned, because this is adhesive, the same type, this type of bioadhesives will be there. This will absorb the inner parts of the body system, and it will release the drugs. The definition mucoadhesive means this is muco, and whereas this type of vehicles, it will act as a adhesive. So we have mentioned this as a mucoadhesive. So body inner layer contains mucosin and water. So this wet surface is using this wet surfaces, this concept will be worked. And these are the protection to inner layers, act as a lubricating agents also. And this mucin secreted by goblet cells. Mucus also contains proteins, lipids, and mucopolysaccharides, and hence can be used as a drug delivery system for targeted and control release systems also. This will be this polymers which are using this in this technology is will be having bioadhesive in nature and adhere to the biological specimen. And mucoadhesion also, it is mucosal membranes that adhere to the mucosin membrane. So it is also having a bioadhesion, and bioadhesions are used in the same meaning. These are the mucoadhesive drug when it is used in this one. So this is the initial stage. With the time, it is absorbing the wet, whatever the present in the moisture or water in the form of water, it is absorbed, and then it will swells and will release the drug in the controlled manner. This will this can be kept as long as we need. Suppose normal systems, if you inject, you cannot take it out. In this case, you can remove. If you feel comfort, then you can really remove this type of devices. The root of mucoadhesive drug delivery systems is ocular drug delivery system, buccal drug delivery system, sublingual drug nasal drug delivery system, gastrointestinal drug delivery system, vaginal and rectal drug delivery systems. You can be used this type of mucoadhesive system, because this most of the parts will be having muco internally, and then because of this property, you can use this type of devices. So mechanism of this is having two stages. One is contact stage and another is consolidation stage. So when we inject this type of dosage, so it goes to the mucous layer and it will get contact, and with the time, because of its adhesive nature, it will penetrate into the mucous layer, mucous membrane, and then it releases the drug. So it will contact, here it will contact, and whereas it will penetrate this area. And swelling of this formulation will give activation to formulation. This can be used in a ocular and vaginal drug delivery system. System is mechanically attached. Even in this type of systems can be mechanically also inserted. And through nozzle drug delivery also it can be inserted. Systems attached by aerodynamics also possible. And gastrointestinal drug delivery system can also be used this type of system. And systematic attached by peristaltic motion can be present in gas. Hence, it can be used in these areas also. Advantages and disadvantages. Advantages with this is less dosage is required and high efficacy will be possible. So less side effects and reduced toxicity will be there because of this mucoadhesive system, and avoids first pass metabolism, and the drug remains at a tissue for a longer time. Hence, treatment will be given for a longer time, and the recovery rate may be improved. In case of infection, drug will reach only to the microorganism, not at all the human body. So disadvantages also, wherever you go, there is an advantage and disadvantage. In this case also, there are some disadvantages of the system. But with technology and focusing on research into this type of, you can decrease this type of disadvantages. chances of immune system reactions can carry using the drug for targeted sites, and needs high sophisticated technology as I mentioned earlier. So high manufacturing cost and time, this also can be avoided by improving the manufacturing, improving the technology. And the difficulty to maintain stability of dosage form. Examples, releasing of erythrocytes are needs 4 degrees centigrade variation. And difficulty to predict or fix the dosage regime also is one of the disadvantages. These are some of the mucoadhesive drug delivery systems advantages. Examples of mucoadhesive polymers is hydroxypropyl methyl cellulose, carbapol, polyacrylic acid, chitosan, polycarbophyl, and hyaluronic acid is a naturally occurring biopolymer with the excellent mucoadhesive properties. This can be used in ocular and nasal drug delivery system also. And this type of polymers will be selected based on their compatibility with the target. Suppose if you are selecting any polymer for this type of targeted delivery system, it should be compatible with that specific tissue or part of that body. Hence, it will be effective. So, while formulating this type of systems, we have to study their compatibility, biocompatibility. And the desired release profile can be set for this one, and specific therapeutic applications can be used. And coming to the latest technologies in polymer matrices, because actually I'm focusing on mainly polymer matrices. So now I would like to tell what are the latest technologies research is going on in this area is 3D printing for customized drug delivery system. It is developed recently. Last time one of my presenter have given about this 3D type printings. This will be used in by fusing fused deposition modeling. The application of 3D printing is to design polymer matrices with whatever the required shapes we want, whatever the shapes is required, and whatever the specified release profiles is required, accordingly we can modify by using this 3D systems. This will help in for personalized drug delivery systems also, depending upon the each individual patient's requirements, such as if the patient is required control release of drugs, or if he is having a chronic diseases, or if we want immediate releases, accordingly we can frame these formulations for to release the acute drug for acute conditions also. And multi-layer 3D printing also, it is nowadays is focusing to improve the polymers with multiple polymers to create multi-layered matrices that can release, because each individual polymer will be having different properties. So in this case, they are selecting multiple polymers depending upon the polymer requirement and the properties of the polymer. So this step may be, as of now, most of the people are using one or two polymers, but nowadays multi-polymers also they are working to improve the systems. For example, a burst release followed by a sustain. Suppose if the immediate response is required, immediate relief is required, burst release, and then again for the long time it has to give me this type of 3D printing will be useful. And the polymeric prodrugs, these are the drugs that is conjugated with the polymeric backbone. In these cases, drugs will be conjugated and attached at the polymer backbone, and it will be activated under specific conditions like enzymatic conditions or pH or light, depending upon the conditions we can modify accordingly. So this approach will enhance the pharmacokinetics and bioavailability of the drugs, preventing the premature release of or degradation before reaching the target site. The self-healing hydrogels also they are working, which will help in to create the drug delivery system that recover their integrity after damage. So once it is injected, it will release the drug and then integrated. Suppose again if we give injection through mechanical or through mechanical stress, again it will activate, and then it will continue to release the drugs. This type of systems also nowadays they are working. And the polymer product system, in this system the drug is conjugated with 2 or more polymers, then at the back of the bone, and is activated under specific condition. This I already explained. And nowadays, artificial intelligence is everywhere. It is, though it is in the initial stage, it has gone through village level also. It has gone artificial intelligence. So in the present case, this drug delivery applications also, this artificial intelligence maybe in the future there is a possibility to use these technologies, and formulations may be made according to the artificial intelligence suggestions, and then machine learning can be used in designing the and predicting the behavior of the polymeric release. Suppose if whatever type of release is required, whatever target we have to achieve. So in the future, maybe artificial intelligence may play a key role. So this also can be used by stimulating the interactions between the drug and the polymer matrix and the psychological, physiological environment also. Say AI could maybe optimize the polymer formulations also in the future for a specific therapeutic targets. And one more important technology is personalized polymer-based drug delivery. Depending upon the each patient data like genetic system, metabolism, and other parameters, AI can design the customized polymer systems which will match their individual patient needs and requirements, which will in fact improve the effectiveness and minimizing the side effects. And this is one of the important upcoming technology, that is nanorobotics and autonomous polymer system. This polymer system will be used in the future for incorporating the polymers into the smart nanorobots, that is at the molecular level, and delivery the drug system at cellular level, even providing the targeted and precise therapeutic actions also. This also system can be used to self-adjust their behavior based on the environmental stimuli and other biological signals, which improves the overall efficiency of the treatment. And the other bio-inspired polymers, incorporating the concepts of biology and such as molecular motors or autonomous transporters may be allowed for highly sophisticated and polymer-based inspired drug delivery system for capable of performing multiple tasks like drug release, sensing, and responding to the tumor environment. So these are the latest technologies which we can expect. Coming to the conclusion, finally we come to the conclusion. I think I have taken more time, so I will conclude with these 2 slides. So for the 2 sessions I have presented, and I will be giving conclusions. Polymer matrices for drug deliveries are evolving rapidly because of the offering solutions for the conventional drug delivery system and challenges with the current available systems. And their ability is also improved to the pharmaceutics due to the pharmaceutical bioavailability and patient compliance also with the sustained and targeted regulatory systems. And as a research and technology progress, the use of polymers in drug delivery systems will be continued to grow, offering even more sophisticated and personalized treatment as I discussed earlier. And it will be option for a wide range of diseases and conditions present in the environmental system. The future will likely see more integrated systems combining personalized medicine, AI, and cutting-edge materials to deliver the drugs with high precision, efficiency, and safety. And as we move forward, the focus will likely to be on smarter, more adaptive polymers, which offer dynamic, patient-specific, and responsive drug delivery system solutions may be possible. So future of polymers in drug delivery application lies in smart and responsive systems that can react to the changes into the biological environment. This system may include the polymers that respond to external stimuli such as pH, temperature, and light, other specific environmental stimuli, and release the drugs in controlled manner. Additionally, advances in nanotechnology and bioengineering are expected to further enhance the precision and efficiency of polymers-based drug delivery systems. With this, I conclude my session. If any questions, please let me know if I could be able to answer.
Thank you, sir. So now it's vote of thanks by Dr. Kasumi Madam, sir.
Good afternoon, sir. Good afternoon, everyone. Good afternoon, sir.
Good afternoon, sir.
I'm
Good
Dr.
afternoon.
Kasimbi. On behalf of Gates Institute of Technology, I take the pleasure to express our heartfelt thanks to Dr. Venkata Prasad, sir, for sharing your valuable insights on synthesis and characterization of polymeric matrices for pervaporation applications, sir. We appreciate the time to you to take the share your knowledge with us, sir. We are very grateful, sir. Thank you so much, sir. Thank you.
Thank you, ma'am.
Awesome.
Thank you, one and all the audience for your patient listening.